Um, Welcome everyone to today's departmental seminar. I am Mark van Iersel, and I have had the pleasure for the last few years to work with Ruqayah Bhuiyan. Ruqayah originally is from Utah, but she has lived in Athens for a long time, and she was an undergraduate in our department. As part of her undergraduate degree, she actually did an internship at Kennedy Space Center in Florida, something that turned out to be not only a great experience, but ended up landing her a job. More info on that in just a minute. So when she worked at Kennedy Space Center, she got really interested in controlled environment agriculture. So after coming back to Athens, she started working in my lab as an undergraduate. We started talking about graduate school. And Ruqayah was a little hesitant because she was actually pretty keen to move out of the Athens area and spread her wings. But when she figured out the benefits of the double dog program, which would allow her to get a master's degree fairly quickly, she decided to stick around a little bit longer, and she is the first double dog in our department. As far as I know, still the only one. Ruqayah successfully defended her thesis yesterday, and she will be graduating very soon. And then if all goes well, thanks to a contact she made while working at Kennedy Space Center, in May, she will be moving to Ontario, Canada to become the grower at a brand new vertical farm there. So we'll all go visit her in summertime and not so much in wintertime. Ruqayah, with that, if you would like to take over and go ahead and present what you have been doing for the past few years.
Awesome. All right, thank you, Mark. So today I will be discussing two studies that I conducted during my time here at UGA, and I'm going to just jump right in. Okay, so to start off, horticultural lighting is necessary for consistent year-round production in greenhouses and plant factories. And this is especially important because in greenhouses, there are seasonal and day-to-day fluctuations in light levels, and in plant factories, there's absolutely no sunlight, so horticultural lighting is absolutely essential. Now, this, it is important, but it can cost growers in the United States upwards of $600 million on electricity alone, so it is important to find different ways to mitigate and decrease the costs associated with horticultural lighting. One way to do that is to use LED lights. One, they use a lot less energy compared to other forms of horticultural lighting, but they're also easy to control. So you growers can control when they apply horticultural lighting, as well as intensity, so how much light that they want to apply, and the spectra of light, so different colors or ultraviolet or far red light. So some key terms to familiarize ourselves with is first PPFDs, or photosynthetic photon flux density, and that is the number of photons that are landing on a square meter area per second, and that is how we measure light intensity. And this is so is because photosynthesis is a quantum-driven process. Now, the amount of light that is landing on a particular plant throughout the day is measured using the daily light integral, and the units for that is moles per meter squared per day. So the first study that I conducted looked at lettuce growth under fluctuating PPFDs. So as I mentioned before, supplemental lighting or horticultural lighting is very costly. And a piece of information that we used to sort of formulate this study was that electrical companies can have variable electricity costs, and that is dependent on demand. So what we wanted to know is, how do fluctuating light levels based on pricing affect plant growth? Here's some background on variable electricity pricing. Variable electricity pricing provides a means of energy management for the controlled environment sector, which is the second highest energy user behind data centers. Now, controlled environment can, as I mentioned before, growers can apply light when they need and at different intensities. So since companies offer variable electricity pricing, it makes it easy to potentially reduce costs. So variable electricity, so as demand increases, so do the prices, and when demand decreases, prices decrease. So can growers provide lighting during off-peak times, which could potentially reduce costs for them, but also lower the strain on electricity goods for the utility companies. This making it a win-win situation. For this study, we wanted to know, how do plants respond to these fluctuating lights? Our objective was to determine the growth effects due to fluctuating lights and understand the physiological reasons for differences in growth. We hypothesized that a constant PPFD will result in more photosynthesis and increased biomass production compared to fluctuating lights, or PPFDs. Now, our hypothesis is derived from the photosynthetic light response curve that as PPFD increases, we see that photosynthesis increases asymptotically. So at higher PPFDs, we would expect higher rates of photosynthesis. For this experiment, we used 2 cultivars of lettuce, Little Gem, which is on the top left, and Green Salad Bowl on the bottom right. We had 6 different lighting treatments, 3 replications, and a randomized complete block design. And our climate conditions are listed below. Here's a breakdown of all 6 of our treatments. Our treatments would go for a high 15 minutes at high PPFD and then fluctuate to a low PPFD after that 15 minutes. On the left-hand side, we have our high PPFDs, and on the right are low PPFDs. We ran our study for a 16-hour photo period, and our DLI, or the daily light integral, was approximately 11.5 for all of our treatments. So as our hypothesis said, we expected that our 400 0 micromole treatment, so fluctuating from a high of 400 to a low of 0, would be our worst treatment, and our best treatment would be our constant PPFD treatment of 200 micromoles. And we would use that constant PPFD treatment as a control and compare the rest of our treatments to that. We grow our plants in a walk-in growth chamber. Here's the setup. This is an overhead of our 3 shelving units, which were our 3 replications. Here is 1 shelving unit, an image of it. We have 6 of our different treatments. And in the red circle, you can actually see that this is our 400 0 micromole treatment, and it's at its low fluctuation of 0. The lights are completely off. Here's just a visual of all of our treatments with our both cultivars of lettuce and our light bars. So the data we collected for this study was first projected canopy size, and this is a non-destructive method of determining plant growth at certain time points during the growing period. And that is done by placing plants in a blackout growth tent and then exposing the plants to blue light. And using a camera, we take images of these plants as they fluoresce. And based on that, we can determine how many pixels are in that image and then calculate the canopy size based on that. Next, we took a net photosynthesis, leaf length, width, and number, the chlorophyll content index of the plants using a chlorophyll meter, the dry mass, the leaf area using a leaf area meter, and then we calculated specific leaf area, which is calculated using dry mass and leaf area, and that helps us determine or give us an understanding of how thick the leaves were. So here, first we are going to look at projected canopy size. Here's just overhead the images that we took. So we would determine canopy size based on that. So in our graph, we have projected canopy size on our y-axis, and then our different PPFD treatments on our x-axis. And then each of the symbols represent different time points. So at 16 days after planting, we noticed no treatment effects. Then we started to see different treatment effects at 23 days after planting, and notice that our 400 micromole treatment had smaller canopies in both cultivars compared to the rest of the treatments. We also noticed that our 360-40 micromole treatment started showing some treatment effects in our green salad bowl lettuce, and it was slightly lower than the rest of our treatments. We also noticed that green salad bowl, which is on the right-hand side, was more sensitive than Little Gem, and that is consistent with the growing habits of this particular lettuce because it is a loose leaf lettuce and Little Gem is a head-forming lettuce. Here is our leaf number, leaf length, width, and chlorophyll content index for both cultivars. The symbols represent the different cultivars. Closed circle is Little Gem and open circle is green salad bowl. And we saw an asymptotic increase in all measurements from 0 to 200 micromoles. And again, we see that our 400 micromole treatment consistently has smaller values compared to all the rest of our treatments. Green salad bowl, which is our open circles, has lower leaf number overall, but larger leaves overall. And this is, again, consistent with growth habits. We also noticed that Little Gem had higher chlorophyll content compared to green salad bowl. Here is our leaf area, dry mass, and specific leaf area. Again, specific leaf area is calculated using our leaf area and dry mass data. Again, asymptotic increases in leaf area and dry mass. And here, again, we notice that at the top, you can see that the leaf area and dry mass for our small fluctuation treatments are pretty similar to each other and are similar also to our constant PPFD treatment. This is early indication that lettuce can tolerate some degree of fluctuating light levels. So we also noticed from this data that Little Gem has a higher leaf area and dry mass compared to green salad bowl. And then we saw an exponential decrease in specific leaf area and Little Gem having thicker leaves. So a higher specific leaf area means thinner leaves, and a lower specific leaf area means thicker leaves. Our 400 micromole treatment has smaller values overall in leaf area and dry mass, but then again, opposite in specific leaf area. So for that particular treatment, we saw that one, the dry mass and leaf area were really small, but then the leaves were actually very thin, so they were trying to compensate for that lack of canopy. Here are some visuals of what our plants look like. This is Little Gem, and this is our 400 micromole treatment, our constant PPFD treatment. You can see that the 400 micromole treatment is a lot smaller compared to our constant PPFD treatment. They're also a lot more yellow and a little spindly. Similar treatment effects were seen in our green salad bowl lettuce. Next, we looked, our next data is going to be our photosynthetic data. And here are all of our treatments. On the y-axis, we have net photosynthetic rates, and on our x-axis, we have time. This is photosynthetic rates for each of our fluctuations, 15 minutes of our high and 15 minutes of low. And in each graph, you can see the values indicate what treatment this is. We saw rapid increases in photosynthesis and stabilization for our small fluctuation and constant PPFD treatments. Then we saw a delayed stabilization in photosynthetic rates in our 360, 40, and 400 0 micromole treatments. We noticed that our 400 0 never reaches photosynthetic rates above 6 micromoles compared to all the rest of our treatments. So in the next set of data, we actually took our data points from our whole 30-minute period and we averaged them. So we looked at our the 15-minute period, so we averaged the values in our 15-minute intervals to see how the plants were responding during each fluctuation. And next we looked at average values over the whole 30-minute period to see how the plants were responding during the whole growing cycle. Here's our data for our average net photosynthesis for the 15-minute intervals. We have average net photosynthesis on our y-axis and PPFD on our x-axis. So as net photosynthesis increases from -1 to 14 micromoles, we saw as PPFD increased from 0 to 320. And we saw a peak and then a rapid decrease in net photosynthesis at even higher PPFDs. We also noticed that our 400 micromole treatment was 65% lower than our at 320 micromoles. Now this is not what we expected because based on our photosynthetic light response curve, we would think that high PPFDs would mean increases in net photosynthesis, but in this case, because our fluctuations were so extreme, especially for the two treatments 400 and 360 40, we saw decreases in photosynthesis at even higher PPFDs. Here is our average photosynthetic rates over a 15-minute period. And we noticed that there was an asymptotic increase in net photosynthesis as fluctuations decreased, which was similar to our growth parameter data. Again, this is a good visual that our 400 micromole treatment had some issues with rates of photosynthesis, and values were a lot lower than all the rest of our treatments. We saw a linear increase. Next, we compared photosynthesis to shoot dry mass and chlorophyll content index, and we saw a linear correlation between those two parameters and photosynthetic rates. And again, our 400 micromole treatment had smaller dry mass and lower CCI compared to all the rest of our treatments. And this is so because chlorophyll biosynthesis is light dependent, and when there's limited light, then there's less chlorophyll, and that affects rates of photosynthesis and ultimately biomass accumulation. Again, all of these small fluctuation treatments were very similar to our constant PPFD treatment. Again, another indication that lettuce can tolerate some degree of fluctuating lights. We then compared chlorophyll content index net assimilation over the 30-minute period by specific leaf area and saw a negative correlation between those parameters, and this might be due to leaf thickness. As fluctuations increased, leaf thickness decreased, and thicker leaves might be able to photosynthesize more because they have more chlorophyll per unit leaf area. Lastly, we compared dry mass to our total dry mass at harvest to our projected canopy size, and As I mentioned before, projected canopy size can be used to track treatment effects and growth during the growing cycle without destructing the plants. And we saw that there was projected canopy size predicted treatment effects at 23 days after planting. So in conclusion, lettuce can tolerate a wide range of fluctuating lights. Extreme fluctuations like our 400 0 micromal treatment, and to a lesser extent our 360 40 micromal treatment, had lower rates of photosynthesis in smaller plants overall. This study has also been published in Frontiers in Plant Sciences. So some practical implications for this study is that tolerance suggests that adjusting to variable pricing is possible, and this could potentially decrease costs for growers, but also decrease the grid strain, which is important for utility companies. And finally, this research is being continued by researchers in the Department of Engineering who are using Raspberry Pi microcomputers to adjust to variable electricity pricing in a greenhouse setting. So we will see how those results come out. Does anyone have any questions or comments? I don't think there are any questions in the chat. Okay.
Hi, Ruqayah.
Yep, hi.
This is Juan Carlos Diaz from Tifton. How I enjoy your seminar. Nice work.
Thank you.
I'm just curious. Just clarify to me, what do you think is going on? It looks like you may have differences in photosynthetic efficiency, right?
Yes.
As affected by how you apply lighting.
Yes.
What may be causing those changes in photosynthetic efficiency?
So, so in our data, we noticed that extreme fluctuation. So at low PPFDs, there's sort of an extent that where photosynthetic apparatuses can continue to be activated. So I think at the extreme fluctuations, those photosynthetic apparatuses are not activated, and so it can't continue to create products necessary for, say, biomass accumulation.
Okay.
Does that answer your question?
Yeah, that's okay. Thank you so much, Ruqayah. Nice work.
Yeah. Thank you.
Hey, Ruqayah, Dr. Reuter here.
Hi.
Good job on your presentation. I was noticing your methods and materials. You had your CO2 about double of what it is terrestrial right now. Is there a reason for that?
I think that just what we mentioned in our paper was that with horticultural lighting, it just increases the efficiency or the that applied light when the CO2 is higher.
Okay, makes sense.
Yeah.
Okay,
and this was also in a growth chamber, not in a greenhouse. So we weren't. It was applied CO2.
So you were adding CO2 to your chambers.
Yeah.
Yeah. Okay. Thank you.
Yeah. Anyone have any other questions? Then I will move on to my next and final study. Okay, so the next study that I conducted was looking at the morphological and genetic effects of far red on lettuce. The two main questions that I asked was, can supplemental far red light increase the amount of captured light? And then what genes are involved in increased light capture? This study was in collaboration with Reeve Legendre and Dr. Savithri Nambison helped with the RNA extraction for gene expression analysis. So some intro, photosynthetically active radiation is commonly defined, not necessarily correctly, as the light spectra between 400 and 700 nanometers. And that often leaves out ultraviolet and far red light, especially with regards to lighting fixtures. And that means that far red is one not included in those lighting fixtures, and more importantly, just underutilized. So there are many studies that look at the morphological effects of far red, but very few look at the genetic effects. And this is what this study focused on. So some big pictures. Far red may upregulate genes associated with leaf expansion, which drive either cell division or cell expansion. And this is important because larger canopies can increase photosynthesis. So if applying far red can increase the canopy size of plants, that could potentially increase photosynthesis and ultimately biomass accumulation. And lastly, I think this study is focused on emphasizing that far red is important in horticultural lighting fixtures. So the first part of this study looked at the morphology of these plants under far red, and our objectives was to determine the morphological and physiological effects of far red, then also understand the genes that might be involved in these changes. We hypothesized that plants receiving the highest amounts of far red will have larger leaves compared to plants receiving less far red light, and genes involved in leaf expansion could be upregulated when far red is applied. For this study, we used green salad bowl lettuce, which is pictured right there. We had 3 far red light treatments of 5, 15, and 30 micromole that were added to supplemental light, and then we had 12 plants per treatment, and we grew these in a reach-in growth chamber. Here is a picture of our growth chamber, and this is an overhead visual of it. We had our 3 treatments that were separated using dividers, 5, 15, and 30. We had our light bar and our 12 plants in each of the treatments. The data we collected for this study was projected canopy area, again using the setup that I explained before, leaf area using a leaf area meter, dry mass, total intercepted light, which was calculated using our projected canopy area data. So over time, we could calculate how much light the plants were actually receiving. Net photosynthesis, leaf length, width, and number, specific leaf area, again calculated using our leaf area and dry mass data. And then the light use efficiency, which is calculated using our biomass and the moles of light that the plant was receiving. So here is just how the plants looked from our projected canopy size calculations. This is at 8, 15, and 22 days after planting for each of our lighting treatments. These are the results that we got. We saw a linear increase in projected canopy area as at 26 and 33 days after planting, but we did not notice any differences at the time points before that. Here is our leaf number data as far red. We have leaf number on our y-axis and far red amount on our x-axis. As far red levels increased, we saw that the number of leaves decreased. And plants in our 30 micromole treatment had the least amount of leaves compared to our other treatments. Here's our leaf length and width data. We saw asymptotic increase in leaf length and linear increase in leaf width. Leaf length and width were highest at our 30 micromole treatment, which is opposite to what we saw in our leaf number. Here is leaf area. Saw an asymptotic increase in leaf area as far red level increase. Far red levels of 15 and 30 had similar leaf areas, despite the opposite trends of leaf number that we saw before. This is our dry mass data, which showed similar trends to our leaf area, saw an asymptotic increase. So as far red level increase, we saw increase in dry mass. This is projected canopy size and dry mass. As I mentioned before, projected canopy size can indicate if there are early treatment effects during the growing cycle. And in this case, we only saw treatment effects at the lapse time point in correlation with dry mass. For a specific leaf area, which is an indicator of leaf thickness, we didn't see any differences between the treatments, but we also noticed that the data that we collected was highly variable, and that could be cultivar or cultivar specific. This is our net photosynthesis data. Again, no differences in net photosynthesis as far red level changed. This is our total intercepted light, which again is calculated from our projected canopy size data. So a linear increase in total intercepted light as far red level increase. And that makes sense because as our leaf area data indicated, as far red level increase, our canopy size increase, and that would correlate with the amount of light that was intercepted by those plants. Our light use efficiency, again calculated using biomass and moles of light the plant was receiving, was not different, so the plants were not utilizing light differently. So next we looked at gene expression analysis, and our objectives for this part of the study was to analyze transcript levels of leaf expansion gene XTH in each of the different far red treatments. We hypothesized that the application of far red would increase the expression of those genes, XTH, and thereby increasing cell elongation and cell expansion. A little bit of background about XTH. It is a cell wall loosening enzyme and can potentially increase cell wall expansion and elongation. And based on that, we hypothesized that it might be upregulated due to far red light. Our hypothesis came from a study by Wagstaff et al. That showed there was high expression of XTH in young leaves. So this is the data that they had. Each of the bars is a different transgenic line, but we're gonna focus on the dark bars, which is Valeria, their wild type cultivar lettuce, and at old and mature leaves, which are the first two grouped bars, we see high expression of XTH at both leaf ages. Now, this might be important for early light capture, because if far red upregulates these cell wall loosening genes, that could encourage the expansion of these leaves and allow the plants to capture more light. So for sampling, we took old, mature, and young leaves, and we extracted RNA to analyze transcript levels of XTH8, and then we normalized the expression levels to actin. Here are our results for our young, mature, and old. We didn't see any significant differences between the treatments or or when comparing treatments and leaf age, but we did see significant differences between leaf ages. So in conclusion, far red light induces leaf expansion. Plants in our 30 micromal treatment had the largest leaves compared to the other treatments. Overall, higher levels of far red are better for light capture, and XTH8 is not upregulated with increased far red, but there might be other genes that might be involved, like cell division genes such as cyclin or KIP-related proteins, or other cell expansion genes like expansin, or other genes within the XTH family. Does anyone just want to say thank you to my committee and my lab members and all of the people who supported me. Does anyone have any questions? All righty. Well, thank you guys for attending my exit seminar.
No questions at all? Or are we just being shy?
That's okay, be shy. Just kidding.
Well, if there are no questions about the second study, then thank you all for attending today, and thank you, Ruqayah, for being a part of the lab for the past few years.
Thank you. Thanks, everyone. I hope everyone has a lovely rest of your day.